So, synaptic delay slows the transmission of nerve signals, undoubtedly. So the more synapses there are in a neural pathway, the longer it'll take for information to get from its origin to its destination. So you might wonder why the nervous pathway is not indeed a continuous wire. After all, the synapses are not due to limitations in nerve fiber length. We have nerve fibers that run from our toes to our brainstem. And we also know that some cells communicate through gap junctions much more quickly than they can communicate through chemical synapses. So why? The reason lays in processing the information, storing it, and making decisions. The chemical synapses are the decision-making devices of the system. The more synapses a neuron has, the greater its information processing capability. Right now, you're using certain pyramidal cells in the cerebral cortex to listen, read, and understand this lecture. Each pyramidal cell has about 40,000 synaptic contacts with other neurons. That means there's a lot of communication going on. And in the whole cerebral cortex, which is the main information processing tissue in your brain, there's an estimated 100 trillion synapses. If you tried to count those at a rate of 2 per second and you were immortal, that would take you about 1.6 million years with no sleep. The ability of your neurons to process information, store and recall it, and make decisions is called neural integration. And as you might be beginning to understand, with 40,000 synaptic contacts and 100 trillion synapses throughout the whole cortex of the brain, it's virtually impossible to understand exactly how this all works. But we can get some good general understandings. Let's look at postsynaptic potentials. Neural integration is based on the postsynaptic potentials that are produced by neurotransmitters. Remember that the typical neuron has a resting membrane potential of about negative 70 millivolts and a threshold of about negative 55. Excitatory postsynaptic potentials are any sort of signal that causes a voltage change in the direction of threshold and makes the neuron more likely to fire. Usually this results from sodium flowing into the cell and canceling some of the negative charge inside the membrane. Glutamate and aspartate are excitatory brain neurotransmitters that produce EPSPs, excitatory postsynaptic potentials. Inhibitory postsynaptic potentials are exactly the opposite. It's any voltage change away from threshold that makes the neuron less likely to fire. The neurotransmitter causes hyperpolarization or further away from threshold, making it more negative and then less likely to fire. This is produced by neurotransmitters that open ligand-gated chloride channels. This causes the inflow of chloride, which is negative, and thus makes the internal environment of the cell more negative. Neurotransmitters like glycine and GABA that you've already explored produce inhibitory postsynaptic potentials, or IPSPs. Acetylcholine and norepinephrine, again, are excitatory in some cells and inhibitory in others, depending on the receptor type of the postsynaptic cell. Acetylcholine excites skeletal muscle, but inhibits cardiac muscle due to these different types of receptors. One neuron may receive input from thousands of other neurons. Some of the incoming nerve fibers may produce EPSPs, while others produce IPSPs. The neuron's response depends on whether or not the net input is excitatory or inhibitory. That is, if the EPSPs override the IPSPs. Summation is the process of adding up postsynaptic potentials and responding to their net effect. This occurs in the trigger zone at the axon hillock. The balance between EPSPs and IPSPs enables the nervous system to make decisions. Is it really time to fire? Temporal summation occurs when a single synapse generates EPSPs so quickly that each is generated before the previous one fades away. This allows the EPSPs to add up over time to reach a threshold voltage that eventually triggers an action potential. In spatial summation, EPSPs from several different synapses add up to a threshold at the axon hillock. So they arrive at the same time, but in different areas of the neurosoma. Several synapses will then emit enough sodium so that the cell reaches threshold at the axon hillock. The presynaptic neurons cooperate to induce the postsynaptic neurons to fire. Here's a figure that demonstrates both temporal and spatial summation. In temporal summation, we're thinking about one neuron, and it elicits several different action potentials, but in such fast sequence that this neurosoma of the postsynaptic cell never returns all the way to resting potential. Eventually, we reach the threshold potential and the postsynaptic neuron fires. In spatial summation, we'll have several different cells adding signal at the same time. But because each cell is adding a little bit, they will summate to produce a postsynaptic action potential. Here's temporal summation of EPSPs shown graphically. One arrives, one arrives, one arrives, one arrives. We finally reach threshold. Boom! Action potential happens. The neurons will routinely work in groups to modify each other's action. Facilitation is the process in which one neuron will enhance the effect of another. The combined effort of several neurons facilitates firing of a postsynaptic neuron through spatial summation. We also might see presynaptic inhibition, which is a process by which one presynaptic neuron suppresses another one. It's the opposite of facilitation. It's used to reduce or halt unwanted synaptic transmission. Let's say neuron 1 here releases GABA at the synaptic terminal of this neuron. This prevents the voltage-gated calcium channels from opening in the synaptic knob, and thus, even though we got a signal arriving at the synaptic knob of the presynaptic neuron, calcium channels can't open, and thus the vesicles of neurotransmitter will not be released into the synaptic cleft. This is presynaptic inhibition. The nervous system must interpret and pass along both quantitative and qualitative information about its environment, whether the light is dim or bright, red or green, whether it tastes mild or intense. Considering the complexity of the information being communicated about the conditions in and around the body, it's a marvel that it can be done in the form of something as simple as action potentials. The way that the nervous system converts information into meaningful patterns of action potentials is called neural coding, or sensory coding, when it occurs in the sense organs. Qualitative information depends upon which neurons fire. Labeled line code is based on the fact that each nerve fiber to the brain leads from a receptor that specifically recognizes a particular stimulus type. Nerve fibers in the optic nerve, for example, carry signals only from light receptors in the eye. These fibers will never carry information about taste or sound. Thus, the brain interprets any signal in those fibers in terms of light, even if the signal results from artificial stimulation of the nerve. You see this effect when you rub your eyes. Quantitative information about the intensity of the stimulus is encoded in two ways. Different neurons have different thresholds for excitation. Stronger stimuli causes more rapid firing rate. An excitement of sensitive, low threshold fibers gives way to excitement of less sensitive and high threshold fibers as the intensity of the stimulus increases. The more strongly a neuron is stimulated, then, the more frequently it fires. The CNS can judge the stimulus strength from the firing frequency of the afferent neurons. This is an example of neural coding. It's based on recordings made from a sensory fiber of a frog's sciatic nerve. As the gastrocnemius was stretched by suspending weights from it, the stimulus strength and stretch increase the frequency of firing of the neuron. Firing frequency is then coded by messages that inform the central nervous system of the stimulus intensity. So far, we've dealt with interactions involving only 2 or 3 neurons at a time. And actually, neurons function in much larger arrangements called neural pools. They may consist of millions of interneurons concerned with a particular body function. They control the rhythm of breathing or moving limbs rhythmically when walking, swimming, running, flying. Information arrives at a neural pool through one or more input neurons. They branch repeatedly and synapse with numerous interneurons in the pool. Some input neurons form multiple synapses with a single postsynaptic cell. These can produce EPSPs at all points of contact with that cell. Through spatial summation, they make it fire more easily than if they synapsed with it at only one point. You can see that here. Within the discharge zone of the input neuron, that neuron, acting alone, can make the synaptic cells fire. But in the broader facilitated zone that you see here, it synapses still with other neurons in the pool, with fewer synapses on each of them. It can stimulate those neurons to fire only with the assistance of other input neurons. So basically, the neurons in these zones would need input from another neuron in order to get as much summation as you see here in the discharge zone. Basically, that means that this input neuron has a vote as to whether these fire, but it needs agreement in order to have them all fire. Such arrangements, repeated thousands of times throughout the central nervous system, give neural pools great flexibility in integrating input from several sources and making decisions. There are different kinds of neural circuits, 4 principal ones. A diverging circuit is where we have one nerve fiber branching and synapsing with several postsynaptic cells. One neuron may produce output through hundreds of other neurons. In a converging circuit, we have the opposite effect. We see input from many different nerve fibers can be funneled into one neuron or neural pool. It's the opposite of the diverging circuit. In reverberating circuits, we'll see neurons stimulate each other in a linear sequence. But one cell, like here, re-stimulates the first cell and starts the process all over. So if there's signal running through this circuit, then we'll always re-stimulate the initial neuron, so it reverberates. We see this in the diaphragm and the intercostal muscles in the creation of rhythmic breathing patterns. Another circuit is the parallel after discharge circuit. Here, the input neuron diverges to stimulate several chains of neurons. However, each chain of neurons has a different number of synapses, and so they'll have different speeds of transmission through the chain. Eventually, they all reconverge on a single output. The delay in their signals arriving at the output neuron allows the output neuron to fire at different times. Now, there's no feedback in this parallel after discharge circuit. So once the signals from each of these chains of neurons have arrived, the output will cease. This explains why you can stare at a lamp and then close your eyes and you can continue to see the image of the lamp for a while. Such a circuit is also important in withdrawal reflexes, where a brief pain produces a longer lasting output to the limb muscles and causes you to draw back your hand from the danger. You may have wondered as you studied this chapter, how can I remember all of this information? It seems like so much to hold on to. And we've sort of addressed this some during the semester. How do we get the memory, the short-term memory to turn into a long-term memory, so on and so forth? So it seems fitting to spend a little time on this now. The physical basis of memory is a pathway through the brain called a memory trace or engram. Along this pathway, new synapses were created or existing synapses were modified to make the transmission easier. Synaptic plasticity is the ability of synapses to change, and synaptic potentiation is the process of making transmission easier. Kinds of memory we'll look into are immediate, short, and long-term memory, and they correlate with different modes of synaptic potentiation. In immediate memory, we're dealing with the ability to hold something in your thoughts for just a few seconds. It's essential for reading ability. In order to understand the words in a sentence in your textbook, you need to be able to remember the words you read at the beginning of the sentence while you're approaching the end of the sentence. Immediate memory has to do with the sense of the present, of what just happened. It's thought that immediate memory might be based on reverberating circuits, because what we just read hangs around in our brain for just a little while, long enough to take the experience of the present moment and anticipate the next. In short-term memory, we're dealing with situations that last from a few seconds to a few hours. Information stored in short-term memory may be quickly forgotten if we stop mentally reciting it. So as you read the text, this is why we stop repeatedly to diagram or recall things, because just a few minutes ago we heard it. Let's recite it again and again and again, and eventually, after we involve as many senses as possible, we hold on to the memory and perhaps it will be committed to long-term memory. Short-term memories can be quickly forgotten if we're distracted, like we're studying and then our child needs something from us, or the phone rings and we forgot all about what we just read by the time we get off the phone. If you think about calling a phone number you just looked up and you have it in your head, but then you're distracted by how to operate your phone and get it to the dialing function, then you forget what the phone number is. Again, it's thought that this involves reverberating circuits. Somewhat longer-lasting memories, however, probably involves an effect called synaptic facilitation, which is different from facilitation of one neuron by another that we see earlier in the chapter. This form of facilitation is called tetanic stimulation, the rapid arrival of repetitive signals at the synapse. Each signal causes a certain amount of calcium to enter the synaptic knob and makes it more likely to fire. If signals arrive very quickly, the neuron can't pump out all the calcium emitted by one action potential before the next action potential occurs. As more calcium accumulates in the synaptic knob, we trigger the release of more neurotransmitter. With more neurotransmitter, the EPSP in the postsynaptic cell becomes stronger and stronger, and the cell is more likely to fire. Thus, tetanic stimulation facilitates the synapse and makes it easier for the postsynaptic cell to fire. Memories lasting for a few hours, such as remembering what someone said to you earlier in the day or remembering an upcoming appointment, may involve post-tetanic potentiation. In this process, calcium levels in the synaptic knob stay elevated for so long that another signal coming well after the tetanic stimulation has ceased releases an exceptionally large burst of neurotransmitter. That is, if the synapse has been heavily used in recent past, a new stimulus can excite the postsynaptic cell much more easily. So you need less stimulation to recover the memory. Now, in long-term memory, we're dealing with two forms, declarative and procedural. Declarative memory lasts up to a lifetime and is much less limited than short-term memory in the amount of information that it can store. Long-term memory allows you to memorize the lines of a play, or your favorite song, or your anatomy and physiology for the test. Declarative memory is the retention of events that you can put into words, whereas procedural long-term memory is the retention of motor skills, the ability to do something like skiing or playing a musical instrument. Some long-term memory involves the physical remodeling of synapses or formation of new ones through the growth and branching of axon terminals and dendrites. As you learn information, you literally make more synaptic connections. And once those synaptic connections have been made, it's easier to use those pathways again. In long-term potentiation, we'll actually see molecular changes. This involves things like NMDA receptors, which are glutamate binding receptors found in the synaptic knobs of pyramidal cells. NMDA receptors are usually blocked by magnesium ions, but when they bind glutamate and are simultaneously subjected to tetanic stimulation, they expel the magnesium and open to admit calcium into the dendrite. When calcium enters, it acts as a second messenger and leads to a variety of different effects. It can synthesize proteins that physically remodel the whole synapse. It can release nitric oxide, which diffuses back into the presynaptic neuron and triggers still more glutamate release. You can see that in these ways, long-term potentiation can increase transmission across experienced synapses. Remodeling a synapse or installing more neurotransmitter receptors has longer-lasting effects than facilitation or post-tetanic potentiation. Thus, long-term memory. The connections of neurons and creation of experiences in the brain, thus, are really determined by the numbers and types of synaptic connections that we make. The pathways that we use most often are the easiest to use. You can think of this in terms of depression also. When someone is depressed and they continually use the pathways of neurons, those will become potentiated also. And thus the depression continues. Sometimes we have to try really hard to walk down a different pathway. It's sort of like picking a path through the forest that hasn't been walked in years. In order to cross that path, sometimes we need tools. But the tools should only be used for a short time. That is, if you're on antidepressants, it's sort of like having a sickle so that you can chop down the bushes that are covering the path and walk freely. Once you've chopped the bushes, you don't need the tools anymore. In the end of your chapter, there's also information about Alzheimer's and Parkinson's disease. I'm not going to go into detail in this lecture because perhaps someone would like to take either of those diseases on as a final project. You can see now that there's tons of final project information coming your way. It's time to really think about what you would like to present on and turn in your topics.